Many people in the street think that natural selection is all that goes on in evolution, but in fact that's not the case. In all natural populations, there are random events occurring, and these random processes are also an important element of evolution. They feed into our ability to infer the history of life, and they are always an alternative explanation of patterns of variation that we see. And therefore, it's important to understand how those effects arise and what their consequences are. That's what we'll be talking about in this lecture. So why does neutrality arise? Well, as you recall, the critical thing in natural selection is variation in the trait. If that's correlated with reproductive success, there'll be a response. And underpinning that is genetic variation. So there's a connection from genes to trait to reproductive success. And the things that cause neutrality are things that cause that relationship to disappear. At the level of the connection between genotype and phenotype, we see that there are many different genotypes that can produce the same phenotype, the same trait. So all of that genetic variation that can produce the same trait is essentially neutral. If there are many different ways of making a person who is 1.61 meters tall, then all of those different ways are effectively equivalent, and we therefore refer to them as neutral with respect to each other because they don't really have any impact on changing the state of the trait. That happens for 4 reasons. The first is that the genetic code is redundant. We will unpack that. The second is that some DNA is not expressed. The third is that some amino acid substitutions don't produce any change in shape or charge of the protein. So effectively, the protein doesn't change its function when you switch out those amino acids. And finally, because the process of development is canalized, which means that it's buffered so that, for example, we tetrapods always get 5 fingers, or tend to get 5 fingers. You can have a lot of genetic variation in the processes that produce hands without changing the number of digits. So the 5 is canalized. Then, once we get to the phenotype, there are also many different phenotypes that have essentially the same fitness. And that can occur because that particular trait variation happens to make very little difference to reproductive success. It may be that the difference between hazel eyes, gray eyes, and blue eyes makes no difference to the reproductive success of the individual carrying them. And it also happens because the impact on fitness of a change in one trait can compensate for a change in another trait. That means that because fitness is a composite feature, it's not only survival times reproductive success, it's all of the different components of biology that go into making up survival and reproductive success, and they can compensate for each other. And that compensation means that different trait states can actually lead to the same output in terms of reproductive success. So let's take a look in a little more detail about those conditions. Here's the genetic code. One of the triumphs of the 20th century was figuring out how this works. It's redundant, and by that we mean we have 64 triplet codons that have 23 meanings. So we have extra triplets. We have more codons than we have meanings. And you can see, for example, that there are 6 different triplets that will code for leucine, there are 4 that will code for proline, and so forth. And these are color-coded so that you can see which are nonpolar, which are polar, which are positively charged, which are negatively charged, and so forth. So, for example, it might be that substituting a histidine for an arginine might not make much difference to the function of a protein enzyme because they both have the same positive charge. Now, the second kind of genetic effect that leads to different genotypes having the same phenotype is pseudogenes and other kinds of non-transcribed DNA. Pseudogenes are genes that are no longer expressed. And they result from a gene duplication that's followed by the loss of function in one copy of the gene. That function is lost because the pseudogene is not expressed, that copy is not expressed. At that point, all of its nucleotides are free to diverge at random as they mutate. So if you have multiple copies of genes and you're only using one, all the ones that aren't being used are not being expressed in the phenotype, and they are accumulating mutational variation. Now, there is some other DNA that's neither transcribed nor translated, and it is also free to accumulate mutations at random. Some of that is transposons, some of it's viruses who have inserted themselves into the nuclear genome. There's actually quite a bit of that kind of DNA. Now, here we have an example of the evolution of a pseudogene. And we have a DNA sequence which is shared by humans and by chimpanzees. And the duplicated gene in the human is actually part of an olfactory receptor. It's functional in chimpanzees, but it's not expressed in humans. And there is both an insertion and a deletion which has occurred in the human copy. So you can see that just in the last 5 million or so years, since we shared ancestors with chimpanzees, variation in this pseudogene has started to accumulate. Now, many amino acid substitutions are also neutral. And if we look at time estimated from fossils on the x-axis, and we look at the number of differences in amino acids that have accumulated in alpha globin, which is one of the two types of globins that go into hemoglobin. It has 2 alpha chains and 2 beta chains. So if we just look at the alpha globin and hemoglobin, and we look at how many amino acid substitutions have accumulated, we can see that that increases with time to last common ancestor. So this is across most of the vertebrates and goes back about 500 million years. If substitutions had been perfectly neutral, then that would be a steady accumulation and all the points would fall on the straight line. There are some deviations from it, but overall the pattern is strong and clear. The older the time to last common ancestor in the vertebrates, the more amino acid substitutions accumulate. And making that point again, if that is perfectly neutral, we expect a straight line. And we get something very close to it. Now, the fourth possibility of having a separation in the connection between the genotype and the phenotype comes from the canalization of development. That means that there are developmental mechanisms that are limiting phenotypic variation. They resist the tendency of variation that's either genetic or environmental that might perturb development. If you take a human embryo, it takes a pretty large genetic perturbation to keep it from having 4 limbs, or 5 fingers, or 2 eyes, or 1 nose. Those things are developmentally canalized. And people with very different genetic backgrounds, with all sorts of genetic variation, encountering many different environments, still grow up to have that stable set of features. That's what canalization means. In this case, there are genes whose impact on the phenotype has been reduced or completely eliminated by canalizing mechanisms. And those genes are then, like pseudogenes, freer to accumulate neutral variation than genes whose impact is directly expressed. In tetrapods, that is, all vertebrates that have come out of the water and lived on land in the last 350 million years, they have 4 limbs, 2 eyes, 1 mouth, 5 digits. And genetic variation is affecting many things in them, but not the number of those traits. That's canalized. Now, at the level of the phenotype, there's some trait variation that makes very little difference to reproductive success. There are various reasons for this. One simply has to do with the size of the population. it may be too small to establish a consistent correlation with reproductive success. It actually takes a good sample size to get a good estimate of a correlation. Some of the variation may simply be irrelevant. For example, there might be variation in traits that cannot be detected by mates, pollinators, predators, parasites, or prey. For example, let's suppose that we're talking about something that might be involved in mate selection, and that species happens to be colorblind. There could be all sorts of color variation that would then be neutral. Another example is variation that lies within the, quote, normal ranges of blood tests. So for example, people who have total cholesterol levels between 110 and 150, variation between 115 and 118 is probably completely neutral. What about systolic blood pressure or diastolic blood pressure? Variation of 5 or 10 or 15 points within that range is probably not affecting reproductive success, survival probability, and so forth. So some phenotypic variation is quite probably neutral. And then, also now at the phenotypic level, a fitness gain in one trait can compensate for a fitness loss in another trait. And if that compensation is precise or close to it in a small population, the change is neutral. This can happen with any trade-off. Now, we're going to unpack that word trade-off and talk about it in detail, but let's just take a few examples where this sort of thing could be going on. More offspring, but less survival. So some people have a lot of children, but die young. Other people have fewer children per year, but live longer, and by the end of their longer life, they have the same number of children. Those are, from the point of view of evolution, neutral. They got the same reproductive success. More success in fighting for mates, but less resistance for disease. That would be a case where success in mating was actually being paid for by susceptibility to disease, and the result would be very similar. There would be more short-term reproductive success, less long-term survival, and the two could compensate so that over the course of the life, those two types had exactly the same lifetime reproductive success. More resistance to normal infection, but more costs of inflammation, and possibly more autoimmune disease. Same story. For this last one, we know that in the 1918 flu epidemic, the mortality rate was highest in young adults who had the strongest inflammatory responses. So they were well suited to resist a large range of normal infections. They probably did better under those circumstances, but when the epidemic came along, they overreacted. There was what's called a cytokine storm. Their lungs flooded with fluid, and many of them then died of secondary bacterial infections. And here you can see an age distribution from 0 out to 75 years of age here. This is the group of people who encountered influenza, all right? So this is the 1918 flu epidemic. And you can see that people of all age classes, including many young people between the ages of 10 and 15, got flu. But the ones who actually died of a secondary pneumonia peaked at age 25. And that was because their lungs were flooding with fluid and providing a nice habitat for bacteria to colonize. So what are the mechanisms that cause random change? They are mutation, founder effects and genetic bottlenecks, the Mendelian lottery, that is, in sexually reproducing populations, meiosis is a fair coin. It gives equal chances for variants to get into gametes, and variation in reproductive success in populations of any size, not just small ones. So the things that drive genetic drift are here highlighted in red. That is the Mendelian lottery, the fact that there's a coin flip in meiosis that is sending genetic variants at random into one gamete or the other, and variation in reproductive success. That is, the body in which they happen to land could end up having a lot of kids or a few kids. And that's at random. So the mechanisms that cause random change. And this is an important point because often people who object to the concept of evolution say, well, it's underlain by a random process. And it's important to understand the sense in which it is random and the sense in which it is not. So, some sites in the genome have more mutations than others. They mutate more frequently. And that occurs in bacteria, and the sites that are mutating more frequently are the ones that are interacting with antibiotics and with the host's immune system. So those are signals that pathogenic bacteria get, and in response to those signals, essentially what they do is they ignore DNA repair for the portions of the genome where it might be good to have more variation. Then there are transitions and transversions. So a transition is a purine mutating to a purine, a pyrimidine to a pyrimidine. A transversion is a purine mutating to a pyrimidine, or vice versa. So transitions are more common than transversions. Now, these mutations are things that are not producing random changes in phenotype space. Some of them may, but mutations don't produce random changes in phenotype space. And what I mean by that is that it is not equally likely that I will have a mutation that results in, say, an offspring having a different eye color and a mutation that results in an offspring having 6 legs. And that is because we have a basic developmental structure which is preventing all kinds of variation from being expressed. Insects can mutate into things that look like insects, and mammals can mutate into things that look like mammals, but we can't have a mammal suddenly acquiring a pair of wings or something like that. That is very unlikely. So mutations are not random in phenotype space in the sense in which I just described. They are random in a very important sense. There isn't any systematic relationship between the effect of a mutation and the need of the organism. Mutations are random with respect to fitness. Mutations do not look ahead, quote. They're, of course, totally unconscious. It's a mechanical process. They cannot project what might work better for the organism. They just create variation. Okay, what is another mechanism causing random change? Well, there are effects going on in small populations that are important. One of them is the founder effect, which is essentially a sampling process. A few individuals found a new population, and when they do so, they bring only a small portion of the genetic variation of the original stock with them. The Pitcairn Islanders are a good example. Also, we have the genetic diseases in Quebec, Tay-Sachs disease, in the Afrikaners in South Africa, that's porphyria. And in Pitcairn Island, there's an unusually high incidence of genetically caused diabetes. These are all cases in which a small population of founders brought with it a disease which was infrequent in the population from which it came, but was quite frequent in the small group that brought it with them. Then there's the genetic bottleneck. So you have a big population, and then it shrinks down to a small size and goes through what we call a bottleneck. Only a few alleles will make it through that bottleneck because there are only a few individuals carrying genes. So there might be, say, 100 MHC alleles in a large population of mammals. But if you restrict it down to 10 individuals, they can at maximum carry only 20 out of those 100, and very probably quite a few fewer than 20. A good example is the homozygosity of cheetahs, and that's confirmed by how easy it is to do reciprocal transplants among them. You can take a skin transplant off of one cheetah and stick it on another, and the immune system will not reject it because they are so genetically similar to each other that it looks like a piece of self. And then the final effect caused by small population size is that correlations decrease. Things get noisier. And if selection in the large population is real but weak, that means there is a correlation, but it's kind of noisy. And then you shrink the population down, what will happen is that the correlation will effectively disappear. And it's, again, it's a sampling issue. Okay, so it's a bit like the founder effect, except this is not an effect on which alleles are present. This is an effect on what is the correlation between the genetic variation and reproductive success. It gets noisier, and finally, if you have very small samples, that relationship can vanish entirely. Then, the mechanism that runs drift has two parts. The first part of it is meiosis. So in a sexually reproducing population where meiosis is the reduction division that's producing the haploid gametes, that process is a fair coin. That means that the probability, if I have a big A and a little a, one on a chromosome from mother and one on a chromosome that came from father, and I want to know what's the probability that those will get into the gametes, the answer is it's exactly fair. They each have exactly the same chance of getting into the gametes. And that's Mendel's first law of segregation. If there are two alleles at a locus, the probability that one of them will get into a gamete is 50%. Now, this next step is a little trickier. Globally, the average number of successful haploid gametes per parent is 2 over the long term on average. There are just 2 chances, 2 flips of the coin, because on average individuals just replace themselves. This isn't true when a population is growing rapidly. It's not true if a population is crashing. But in the long haul, there really is just replacement. If there weren't, then we would be neck deep in rabbits and hip deep in crocodiles and smothered by elephants. Okay? So on average, population sizes are stable, and that means that in this coin flip, there are really long-term just 2 opportunities to get into the next generation. The other part of the mechanism that runs genetic drift is that the trait or the gene can land at random in families that have different sizes. And that would be true if there was no correlation of the trait with reproductive success. So the gene is not affecting anything that's associated with reproductive success. So if an allele randomly lands in a sequence of families with different numbers of children, then it will go up in the families with lots of children, it will go down in the families with fewer children. And that is actually the meaning of having 0 correlation with reproductive success. And in a case like this, its frequency goes up and down pretty much at random. What does that look like? Well, it produces an aimless wandering of the frequencies of neutral genes. That resembles the Brownian motion of small particles being pushed around by the thermal motion of molecules. It's driven by these 2 factors, meiosis and variation in reproductive success, and it fixes neutral alleles faster in smaller populations, but it occurs in populations of all sizes. So here are some computer simulations of the process. You see 3 panels. The top one is a population of 20. The middle one is a population of 200. The bottom is a population of 2,000. The x-axis is time. The y-axis is allele frequency. In all cases, the simulations were started with 2 alleles at 50% frequency, right in the middle here, and then it was allowed to run as described with meiosis occurring and then random variation in reproductive success. And what you can see is that these populations, each one of these lines is the frequency of an allele, one of the 2 alleles, in one population. So that's one population, that's another population, that's another population. And you can see these populations start to diverge. Things look very noisy in the small population. In a larger population, pretty noisy, but a little bit less. And in a very large population, there's still variation which is accumulating among populations in the allele frequencies, but it takes longer to do so. The result of this essentially is that alleles get fixed. What that means is that if they go to 100%, they can't change anymore because everybody has them. And that process happens more rapidly in small populations than in large ones. Now, what's going to happen with these neutral alleles? Most of the neutral alleles that come in by mutation never really make it to high frequency. They disappear because of drift fairly quickly. And if they do get fixed, if they do go to 100%, it will take them longer to do so in a bigger population. But bigger populations also have more mutations. And if you are counting over the whole genome, this isn't for one locus, but over the whole genome, if you are counting the number of mutations that get fixed in a unit of time, the fact that large populations have more mutations is exactly compensating for the longer time to fixation of any single mutation. Now, we don't know which mutation will be fixed, but we do know how many will be fixed on average if you look at the whole genome. So, in that sense, there is a molecular clock, and that molecular clock is like an atomic clock. An atomic clock is driven by radioactive decay. And in any given period of time, we don't know which atom will decay, but we do know how many will decay on average. And the regularity of this process is a result of the very large number of independent events. Our haploid genome consists of about 3 billion DNA base pairs, and 1 mole of uranium contains about 10 to the 23rd atoms. So we are dealing with very large numbers, and when that huge sample size is there, random events become regular in terms of how many of them are occurring per unit time. If we look at this process in the case of flu, the neat thing about flu is that it has been frozen ever since the 1930s. And we can go back and we can sequence its entire genome. It's a fairly relatively small genome, large for a virus, but very small for an organism. And we can look at how many nucleotide substitutions there have been since the year it was isolated. Now, over this time, population sizes fluctuated. They fluctuated hugely. There were big flu epidemics, and then immunity built up, and the population of viruses became much smaller, and then it came back again. However, look at the regularity in the substitution rate. It's quite linear. In this case, the effect of population size on the number of new mutations that were introduced each generation exactly compensated for the slower rate of the neutral mutations in the smaller populations. So if you can estimate time from number of substitutions, you have a molecular clock. This is a molecular clock. It says that if we had this many substitutions, that was 5 years. So in summary, there are several reasons that variation in DNA and variation in traits may make no difference to reproductive success. Meiosis is like a fair coin. The probability of getting into a specific gamete is 50%. The fixation of neutral alleles is like radioactive decay. In neither case, neither in genetic drift nor with radioactive decay, do we know precisely which mutation will be fixed or which atom will decay. But because there's so many of them, we know how many events will happen on average in a unit of time. Genetic drift fixes neutral alleles at a regular rate that is independent of population size, which means that over the long course of evolutionary history, it provides a very, very regular accumulation of change. And that regularity allows us to estimate times to last common ancestor. So the bigger the difference between DNA sequences, the longer it's been since those two organisms shared a common ancestor.